Hi viewers, in this class we are going to discuss the humoral immunity apart from the types of immunity. Under this topic, we are going to discuss the definition of immunity, types of immunity, definition of humoral immunity, properties and functions of antibodies at a glance, and development of B cells, types of antigens, B cell activation and responses, finally, the mechanism of humoral immunity. As we know, the immunity is the ability of the organism to defend all the substances which are from the foreign resource. Then this immunity is classified based on the type of cells which are involved. There are two kinds of immunities, namely humoral immunity and the cell mediated immunity. Humoral immunity and cell mediated immunity. Humoral immunity, the basic cells which are involved in this are B lymphocytes or B cells, whereas T cells, phagocytes, natural killer cells, they involve under the cell mediated immunity. So in this class we are going to discuss the humoral immunity in detail. See here, there is the involvement of the B cell in the humoral immunity, T cell in the cell mediated immunity. Come to the humoral immunity. Here the immunity mediated by the antibodies that are released into the body fluids such as humors. Body fluids are called as the humors, such as plasma, lymph, etc. is called as the humoral immunity. The immunity mediated by the antibodies that are released into the body fluids such as plasma, lymph, etc. is known as the humoral immunity. And if you observe the properties and the functions of the antibodies, antibodies are the soluble proteins. The activated B lymphocytes, they differentiate into plasma cells. Plasma cells secrete antibodies. Antibodies show specificity against two antigens and globular proteins that provide immunity. So hence the antibodies are known as the immunoglobulins and denoted as Ig, immunoglobulin. Here there is a development of the B cell. Here the B cell finally get converted to effector cell in the form of the plasma cell and the memory B cells. Plasma cell has the ability to produce many antibodies into the body fluids. So this antibody mediated immunity is nothing but the humoral immunity. Here these are the plasma cells and they secrete antibodies. They act against different kinds of pathogens and the disease causing agents. In general, what happens in the secondary lymphoid, this is the production of the antibodies where the B cell is inactive. The B cell is inactive until and unless there is a antigenic stimulation. Prior to the antigenic stimulation, the B cell is said to be as inactive B cell. Upon the activation of antigen, the attachment of antigen to the surface receptors of the B cell get activated or undergo co-stimulation. Right? It maybe takes place through the help of the T helper cells. They activate the B lymphocytes by producing different kinds of cytokines and interleukins. So especially interleukin 2, and thereby these B cells get activated and undergo differentiation into effector B cells, which are nothing but the plasma cells, and the memory B cells. Memory B cells, they kept inside the body for days, months or years together. And whenever the same antigen re-entered into the individual, then they show the secondary response. And whereas the effector cells in the form of plasma cells, they tend to produce antibodies. These antibodies take care of the killing of antigen or neutralization of the antigen. And the functions which are mediated by antibodies, usually opsonization, activation of the complement, and antibody dependent cell mediated cytotoxicity ADCC, and the transcytosis are the basic functions of the antibody. See here, antibody can neutralize the microbes and toxins, opsonization and enhance the phagocytosis by the phagocytes, and ADCC, lysis of microbes, phagocytosis of microbes, and inflammation, complement activation. These all are the functions of the antibodies. And if we see the basics of the humoral immunity, generally B cells are developed from the bone marrow and undergo antigen induced activation and differentiation in the periphery of the bone marrow. What is the activation is, prior to the antigen stimulation, the B cell is in the inactive form. After the antigenic stimulation, it converted into activated B cells. These activated B cells can give rise to antibody secreting plasma cells and the memory B cells. This is nothing but the differentiation of the B cells. The activated B lymphocyte can differentiate it into two forms. One is the effector B cells or plasma cells, and another one is the memory B cells. See here, plasma cells, they are always in alert condition. They secrete different kinds of antibodies and they take care of the killing of antigen. Whereas the memory B cells, they kept aside inside the body of the individual until there is a secondary stimulation of the antigen. Now during the B cell activation, what happens? The sequential immunoglobulin gene rearrangements transform a pro B cell into an immature B cell. This immature B cell expresses IgM antibodies with a single antigenic specificity on the surface of the B lymphocyte, and thereby further development of the B cell yields the mature naive B cells expressing both IgM and the IgD molecules. So this is the maturation of the B cells. And within the lymphoid organs, there was a negative selection and the receptor editing. What is the basic function of the lymphoid organs is activating the lymphocytes, and those which can identify the non-self antigens, they will be selected for the clonal expansion and the clonal proliferation, whereas those which develop the markers, otherwise the receptors for the self cells, for our own body cells, they will be killed off. So the cells with the self-reactive BCR, if the B lymphocyte as a receptor which can interact with the self cells, self-reactive B cell receptor, if it is expressed upon the surface of the B cell, then that will be killed off by the negative selection and the apoptosis, programmed cell death. And what should be happen? The B cell should be developed and they have to identify the non-self cells which are entering into our body every time. So there is a positive selection of the good quality B lymphocytes. Those which develop the self-reactive BCR, they will be killed. Those which develop the non-self-reactive B cell receptor upon their surface, they will be selected for the further development. And The B cells undergo receptor editing to produce non-self reactive membrane bound immunoglobulin. Membrane bound immunoglobulin. And the antigens which are interacting with the B cells are again two types. Some of them they are thymus dependent, they require T lymphocytes to present this antigen, and some of them they are thymus independent, no need of T cell for the antigenic activation or antigen presentation. Here, based upon the presence and absence of the T lymphocyte for the activation of the B lymphocyte, there are two kinds of antigens. Those antigens which are dependent upon the T lymphocyte, they are called as the thymus dependent antigens, and those which do not require any kind of help of the T lymphocyte for the activation of the B lymphocyte, they are called as the thymus independent antigens. Thymus independent antigens. Here we can observe clearly, this is the thymus independent antigen. The antigen comes in contact with the surface receptors of the B cell directly, and thereby there is a signal transduction and gene rearrangement and the production of the appropriate antibodies by the B cell. But here in the second example you can observe that some of the antigens they require the help of the T helper cell. So here this is the antigen, red color one is the antigen. Now this antigen requires the help of the T cell in order to activate the B lymphocyte. So that these are called as the thymus dependent antigens, T cell dependent antigens. Here you can observe furthermore, the T independent antigens are multivalent, like the bacterial polysaccharides are repeating determinants on the surface of the viruses. Responses are fast, that is within 1 to 2 days, and predominantly IgM kind of antibodies are produced. And this kind of T independent antibody production is weak in infants and the young children, whereas T dependent antigens must contain a protein component so that T cell help can be received. Responses are very slower, it requires several days to respond. Produce all the Ig isotypes, immunoglobulin M, immunoglobulin G, immunoglobulin A, and IgE, and the response is very stronger and can lead to antibody affinity maturation and the memory. So this is the T independent antigen presentation and the T cell dependent antigen presentation. And coming to the B cell activation, here there is a attachment of the antigen upon the B cell receptor, attachment of the antigen on the B cell receptor, and the B cell which is activated, it sends a signal. It is the consequences of the signal transduction process triggered by the engagement of the B cell receptor with that of a appropriate antigen, ultimately leading to many changes in the cell, including the changes in the impression of the specific genes, rearrangement of specific genes. In general, B and T cell activation, B cell and T cell activation share many parallel programs, including the compartmentalization of functions within the receptor subunits, activation by membrane associated protein which is called as the tyrosine kinases, the assembly of large signaling complexes with protein tyrosine kinase activity, and recruitment of several signal transduction pathways. The B cell receptors can intensify the activating signal resulting from cross-linkage of membrane Ig, cross-linkage of MIG. This may be particularly important during the primary response to low concentration of antigens. The cell membrane associated molecule CD22 can act as a negative regulator of B cell activation, protein tyrosine phosphatases dock to ITTM immunoreceptor tyrosine inhibitory motif of the cytoplasmic tail of CD22 deactivate the B cell receptor associated signaling complexes by removing the phosphatases added by activating protein tyrosine kinase. Protein tyrosine kinase. Activation induced by if the activation induced by the thymus dependent antigens requires contact dependent help delivered by the interaction between CD40 on the B cell and CD40L, an activated T helper cell, the CD40 CD40L interaction is essential for the B cell survival, the formation of germinal centers, the generation of memory cell population, and somatic hyperstimulation. The CD40 and CD40L combination of B cells and T helper cells is very essential for the formation of the germinal centers, in the generation of memory cell population, and also for the somatic hypermutation. The primary antibody response has a long lag period, and IgM is the first antibody class produced, followed by a gradual switch to other molecules. The secondary antibody response has a shorter lag time and the response lasts longer. IgG and other isotypes are the main products generated in the secondary response rather than IgM, and the average affinity of antibody produced is higher. Germinal centers, that is the sites of somatic hypermutation of rearranged immunoglobulin genes, form within a week or so of exposure to a thymus dependent antigen germinal centers. The germinal centers are the sites of affinity maturation, formation of memory B cells, class switching, and formation of plasma cells. So here you can observe the virus. These are the epitopes are antigens, the B cell, naive B cell activation, that is attachment of the antigen to B cell. So there is a activation of the B cell that leads to the lymphoblast, and this B cell differentiated into effector plasma cell and the memory B cell, effector plasma cell that produce appropriate antibodies. And here we will differentiate the responses into primary response and the secondary response, where primary response is the response when the antigen for the first time come in contact with the B lymphocyte and elicit a response which require more time, and the amount of antibodies are produced in low number. That is the primary response. In the primary response, IgM plays a major role. IgM plays a major role in the primary response. Whereas, if you see the secondary response, if the same antigen, as a result of the primary response, what happened? There is a differentiation of B lymphocytes into effector plasma cells and the memory cells. Memory cells kept aside in the body, and the effector B cells, they produce the plasma cells, they produce the antibodies. The antibodies take care of the killing of antigen. Now, if the same antigen if entered into the body for the second time, the memory B cells identify the antigen, and within short period of time, short span of time, many number of antibodies are produced to neutralize or kill the antigen. During this time, based upon the type of antigen which is entered into the body, there is a class switching of other isotypes. Instead of IgM, there are the production of other antibodies like IgG, IgA, IgD, and IgE. So primary response takes more time and number of antibodies produced is low, and whereas in the secondary response, short span of time it requires and more amount of antibodies are produced against the antigens. And here we talked about the germinal centers. If you observe the secondary lymphoid organs, There are the primary follicles and the secondary follicles. Secondary follicles are enriched with the germinal centers. These germinal centers are the sites of hypermutation of rearranged Ig genes, and these are the sites of maturation of the B lymphocytes and T lymphocytes. Basically here, when we are talking about the humoral immunity, these are the B lymphocytes, and class switching of production of different kinds of antibodies, and the formation of plasma cells and the memory B cells. Here you can observe, this is the somatic hypermutation of the B cells, and here this is the B cell follicle, and this is a T cell B cell border, and these are the long lived plasma cells which are the memory B cells, and here short lived extrafollicular response where we can find out the effector B cells. So these are the germinal centers. Here also you can observe, for the first time the antibody, upon its surface it may contain IgM or IgD as the receptors, IgM or IgD antibodies as the receptors. Upon the antigenic stimulation, here there is a formation of the IgG. So this is a short-lived humoral immunity. This is the extrafollicular response which remains for days. Here follicular germinal center response, it remains for weeks or months. Here there is a class switching of IgM, IgG, and here these long-lived LLPC means long-lived plasma cells, they produce more long-lived humoral immunity. So plasma cells, they secrete IgG molecule which kill the antigen. Finally, the humoral immunity can be inhibited by antibody feedback, binding of antigen antibody complexes by receptors on B cell inhibits the BCR signaling. And coming to the mechanism of humoral immunity, what is the mechanism behind the response of antibody? Here, under the mechanism, we will discuss these things. The free antigen which is present inside the body that binds to the fragment of antigen binding sites upon the surface of the B lymphocyte, and the free antigens bind to the FAB end of the antibodies that are present on the surface of the mature B cells. They engulf and process the antigens, then they display them as the antigen fragments on their membrane with the help of the class 2 MHC molecules. Then appropriate T helper cells recognize them and interact with them. The antigen MHC complex, MHC and antigen complex will be formed and release a type of interleukins which stimulate the B cell to proliferate and differentiate into memory cells and the effector B cells, otherwise the plasma cells. So here the IL stimulates the B cell differentiated into plasma cells and the memory cells. Plasma cell produce specific antibodies. Plasma cells produce the specific antibodies into the plasma or into the they will be released into the extracellular fluids. These antibodies helps in the opsonizing and immobilizing the bacteria, neutralizing and cross-linking of antigens, leading to the agglutination of insoluble antigens. Otherwise, the precipitation of the soluble antigens. They also activate the phagocytes and complement system, and the products of antigen antibody reactions, which are called as antigen antibody complexes or immunocomplexes, they are removed from the body by the eosinophils and monocytes very frequently. This is all about the mechanism of the humoral immunity. Here you can observe the same in the diagrammatic representation. A B cell is triggered when it is encountered its matching antigen, and the B cell engulfs the antigen and processes it and then displays the antigen through the help of the MHC class II molecules. Now, this combination of antigen and MHC attracts the help of a mature and matching T lymphocytes. Thereby, these T lymphocytes they produce different kinds of interleukins, otherwise the cytokines. These cytokines secreted by the T cell help the B cell to multiply and mature into the antibody producing plasma cells. These plasma cells has the ability to produce antibodies. The antibodies are released into the blood. Antibodies lock onto matching antigens. The antigen antibody complexes are then cleared by the complement cascade or by the liver and the spleen. So this is all the mechanism behind the humoral immunity. Humoral immunity made by B cells and their products like the antibodies. Thank you one and all.